Okay, everyone. Well, it is now noon on my clock, so here we are at week 1 of high school biology. We are going to start today with a little introduction about the course. We're going to talk about some lab safety that we will need during the class. We're going to talk about your lab kit briefly, and we are going to talk about classification and organisms. So I see a couple of people in the text chat are writing about their lab kits. What I would like to do is I would like to make sure everyone has their lab kits. So Sarah, you do not have yours yet. Let me just write this down. Gabriella, you have 2, so I will send you a label to return one of them. Alicia, do you have your lab kit yet?
Yes, ma'am.
You do. Fantastic. And then Robert, do you have your lab kit? Robert, do you have your lab kit? Awesome. So, Sarah, I will check with your advisor, and I will make sure that yours is on its way. I believe they were all sent last week, so it should be coming. And, Gabriella, I will work on yours and send you an email or give you a call about that after class today. So we are going to meet each week on Wednesdays at noon Eastern Time until 1:30. This class is a little bit difficult to gauge in terms of time. I am going to do my best to finish by 1:30. However, there may be a few classes while we are doing the dissections where we may run over by a couple of minutes. So my suggestion to you is to just give yourself a little bit of leeway if you're making an appointment or have something to do after class, especially during weeks 5, 6, 7, 8, and 9, when we could be doing dissections, because you will need to clean up for a couple of minutes. Otherwise, your house will really smell. So, high school biology is a half a credit course. You do need to complete all of the assignments. You do need to come to the classes in order to get the half a credit. Your assignments will be given an actual letter grade and a numerical grade. If you turn your assignments in late, you will lose points. All of your assignments are due Wednesdays at 9 a.m. So your homework for next week is due next Wednesday. Or your homework for this week, is due next Wednesday. If you finish your assignment early, turn it in early. Once I get your assignment, you will either get an email back from me or you will get a response from Moodle saying that your assignment has been graded. The current grade is whatever. Any comments that I have, if I ask you to fix something on your lab report or on your assignment and you would like to fix it, you certainly can. Another portion of the grade for this class is class participation. We are going to be doing an activity at least once a week. There are lots of opportunities for you to turn your cameras on and let me watch you do the assignment. I'm going to do that more with the dissections than anything else. But what I really want to see is you guys working. You don't need to listen to me the entire time. So make sure that you participate in class. I will, however, never turn your camera on if you do not want it on. So just keep that in mind. In order for me to turn your camera on and for everyone to see it, I need to click a button on my end, and then you also need to click a button on your end. So don't worry about, oh no, I don't want to be on camera today, I didn't get a shower yet. Or I'm in class in my pajamas, not a big deal. I will never force you to be on camera. However, I certainly encourage you to be on camera as often as possible. In your syllabus, it had listed the labs that we were going to do each week or activities and the labs and activities for homework. Some activities require you to have additional items. Most of the time, those additional items are things that you would find in your house. Sugar, water, spoons, things that are easily accessible. When we do the dissections, you are going to want to make sure that you have paper towels available to you. So just make sure that you look at what you need for class before you sit down for class so that you are prepared for class. We are going to start by talking about lab safety today. Anyone have any questions just about the class in general before we go ahead and get started? Awesome. Alicia, go right ahead.
I looked on my our syllabus in the email, and it says on week 5 to have frozen corn, bean seeds, and celery. It wasn't highlighted or bolded as in if it was something that we needed, but I didn't see any seeds other than the bag of seeds for today in the lab kit.
Okay, well, great question. Well, in this bag of seeds today, there actually are some bean seeds and the corn kernels. So we can use that, but the celery should have been highlighted. I'm not sure why it wasn't. So that was week 5, you said. I will go ahead and fix that. And for the corn, you could use frozen corn. It's going to make it a lot easier to cut through, like, you know, what you would cook for dinner out of the freezer section of your grocery store, or you can use the corn that's in this bag. So I will go ahead and highlight that as well. Now on that note, if you do find that you are not able to get something for the lab, let's say you checked your freezer and there was frozen corn in it the night before, and then you go to use it and your mom has used it in her chili. Don't worry, every lab either I am going to do on my camera or I'm going to ask one of you to do on camera. So anything that is being done in class, if you don't have the materials, you will also be able to see live on camera. It makes it a lot nicer if you're able to follow along with me, but if for some reason something happens, by no means is it the end of the world, and do not get worked up about it. If for some reason you are not going to be able to make class one day, you're sick, you have an appointment, you have no power at your house, if you could just shoot me a quick email, give me a call, just to give me a heads up, that's fine. All of our classes are recorded, and they will be posted on the Moodle site after each class. So if you miss a class for whatever reason, you can always go back and take a look at it on the computer. Or you can refer back to the recordings when you are working on your lab report or for whatever reason. Great question, Alicia. Anyone else have any questions before we go ahead and get started? Okay, well, we are doing a lab class, and as with any lab class, we need to make sure that we are safe in our course. So on your PowerPoint screen right now, you should see a PowerPoint that says Lab Safety on it. I'm going to turn my camera off, and I'm going to make my Lab Safety PowerPoint larger. If you recall from the orientation, the way you make your PowerPoint larger is you look at the purple toolbar right above the PowerPoint. On the end, there is a little white or gray arrow that is pointing up and to the right-hand corner. Go ahead and click on that, and your PowerPoint screen should now take up the majority of your computer screen. So let's go ahead and take a look at lab safety. Even though we are working in your homes and we are not working in an actual lab, there are still several safety precautions that we need to take to make sure that we are safe and that our surroundings are safe. The first thing, the most important thing you can do is to be prepared. So lab safety begins before you even go into the lab. You should always Read through the lab instructions the day before you go to the lab. Today, I'm not worried about this, but for next week, I expect you to read the lab before class starts. If you have any questions or concerns, ask me before we start, either at the beginning of the class or go ahead and send me an email. Everything we are working with in the lab kit is relatively safe. However, if used incorrectly, some of the items in the kit can be dangerous. We should always have proper lab behavior. You should never indulge in horseplay or behavior that could lead to the injury of others. Okay, and here you see two students fooling around in the lab. Now, you are not actually going to be in the lab with other students, but an example of this would be when we are doing dissections. Make sure that you are careful with the scalpel and the pins that we use during the dissections. Fooling around could lead to you becoming injured. Dress for lab. You should use goggles during every lab. If your lab kit did not come with goggles in it, go ahead and either take a pair from your father's workshop or go ahead and buy a pair from the hardware store. They do not need to be fancy goggles, just something to go over your eyes so that liquid does not splash into them. You will not need them for today's lab, but it is good practice to use lab safety glasses during all of your labs. Because that we will be using some glassware and we will be using some corrosive liquids, you should always wear sneakers or at least socks on your feet, no bare feet or no sandals, so that if something were to either spill or fall on your feet, it will not cut or burn you. Know where your nearest water source is if you need to flush chemicals off your hands or out of your eyes. I am going to complete all of our labs on my kitchen island. I suggest that you complete your labs somewhere where there is water nearby. If you get chemicals on your hands, you will need to be able to go wash your hands. Okay? If you're working at a desk in your study, that's fine. Just know where the quickest spot to get to water is. If you were to get a liquid in your eye, you are going to cup your hands and run it underneath the sink so the water flows right over your eye. If you get a chemical on your hands, you are going to go wash your hands. We do not use anything that is in a large enough quantity to require you to go take a safety shower. So we do not need to worry about that. Okay, some general science safety tips. If you look at the man on the right-hand side, he looks kind of like a cartoon character. He's looking at a test tube. That is how you should look at a test tube. You should always look at the side of a container, whether it's a test tube, whether it's a beaker. You never look down from the top into a test tube. Anyone have a guess as to why? Why would we want to not look down into a container? Alicia.
Some chemicals could be harmful to your eyes or to your nose.
Very good. The chemical, either the fumes from the chemical, or there could be a chemical reaction that causes the chemicals to bubble up, and they could be harmful to your eyes, to your nose. Very, very good. Great job. Okay, if you're heating a test tube, you should always point it away from you and point it out in case something were to bubble up. You should never taste anything in the lab. Even though some of the items we will use are edible, none of it should ever go in your mouth. And you should not eat or drink or chew gum during lab. That's just because you don't want to cross-contaminate anything. Okay, we should never smell a test tube directly. Use your hand to fan the odors towards your nose. And if there is an accident in your lab, regardless of how minor you feel it is, please let me know. Okay, if you drop your beaker and it cuts your hand, just tell me. We need to keep a record of any incidences that we have. If you were to cut your hand or to burn yourself, let me know. If you do burn yourself, all you need to do is run it under cold water. We actually will not be using any Bunsen burners in class. However, there are a few labs where we will use hot water, where you'll need either a stove or a microwave to heat the water up. Okay, and at the end of each of your labs, you are going to need to clean your glassware and return it to the lab kit once dried. This is especially important because no one is going to be there to check on you. I'm not going to be around to say, you got to put that away. Everything should be washed after every lab. Everything that we use, chemical-wise, can go down your sink in the amount that we use them. The dissection specimens just go in your trash can when you are done with them. Nothing special needs to be done. There is no special way to handle them. Okay, so that is where we are in terms of lab safety. Anyone have any questions about safety for our lab? Fantastic. Okay, so let's start talking about science. I looked over your assignments, the orientation assignment that you guys had entered, where you wrote what you were interested in most in terms of biology. But we're going to start off with some basic science right now. One of the things that makes science science is the ability to observe your surroundings and be good at observing your surroundings. So what I would like you to do is I would like you to take 2 or 3 minutes, look at your whiteboard. I have unlocked it, so everyone should be able to write in it. And I would like you to make some observations about the room you are sitting in. I'm going to make some squares up here so everyone has a spot to write. And since we all have different first letters of our names, that's how we will distinguish between them. So Alicia, you are... Oh, it's not letting me finish that. All right, Alicia, you are this first square. Gabriella, you are the second square. Robert, you will be the third square. And Sarah, you will be the fourth square. Now, as a reminder, to get to your whiteboards, you are going to look at the bottom right-hand side of your screen. You should see it says Make Your Observation. Make that screen larger. And you should see your square. Then what you are going to do is either click where it says Text or click where it says Text Block, one of the block letter A's on the right-hand side. Then click on the whiteboard, and you can begin typing. I'm going to do my observations right here in the center of the whiteboard. But just give me some observations about your room. This does not need to be a paragraph. I'm not all that concerned with grammar, spelling. Just make some observations. So for example, the rug in my room is green with light blue walls. There is a desk with a chair and a computer. Okay, and you make some observations about your room. Okay, so remember, in order to type, you can click either of the A's where I am drawing an arrow right now. And that should allow you to type on the screen. If you can't get rid of the word text, just typing right after the word. It's not a big deal at all. When you start typing, you should be able to tell me the observations about the room you are sitting in. Don't worry about centering anything in your squares. Just go ahead and type. If it runs out of your square, not a big deal. Okay, I'm going to give you 1 more minute. Finish up your thought. All right, fantastic. Thank you guys very much. It can be very difficult to try to center things, so please don't worry about that. So up in the top one, we have Alicia's. Go ahead and read Alicia's. This will give you a picture of her room. Alicia gives me a good idea of what her room is like and the desk that she's sitting at. Those are great observations. Okay, when I read Gabriella's, again, I'm given a good sense of where she is sitting, the color of the wood, and the tile flooring. And Robert's and Sarah's as well. As I read through them, I am getting a good idea of what is going on and what the room that they are sitting in looks like. Making observations is very important in science. Everything that we do has to do with observing our world around us. Today's lab, we are going to use some observation skills. And right now, we are going to do an observation activity. on your whiteboard, I just put up two pictures. Both are pictures of a giraffe. They're both the same pictures with a few subtle differences. What I would like you to do is to go ahead and on your whiteboard, circle the differences that you see. For example, the coconut on the right does not have a mark, where the coconut on the left does. So go ahead and make some observations. Circle the differences. I used the pencil that I just drew my arrow to, and I changed my color to blue. You can change your color on the bottom of your screen to something else, or you can leave it black. It is up to you. Please, however, do me a favor and do not try to erase something if you draw it incorrectly or in the wrong spot. So go ahead and find the differences between these two diagrams. Okay, it looks like you guys are doing a great job. I think you have them all. I'm just double checking here. Okay, I believe you got them all. Fantastic. So now we are going to go back to our whiteboard, and we are going to talk a little bit about taxonomy. Now, taxonomy is how scientists classify living things using characteristics. So click on your PowerPoint screen for me, which is the top right-hand screen. If I were you, I would go ahead and make the screen larger. And we are going to take a look at the characteristics of living things. First, we have living things are made of cells. That's all living things. Some living things are unicellular, some are multicellular. Okay, and here I put a picture of red blood cells, and onion skin cells, and human cheek cells. There are several characteristics of living things, besides being composed of cells. They also grow and develop. Growing and develop means they increase in cell size and or number. It includes development, aging, and death, and cell differentiation, which means that cells are specialized for certain jobs. Cells in your liver are specialized to do jobs that your liver does. Cells in your brain are specialized for your brain. There are some cells called stem cells that can be used or can change their specialization. Living things also need to obtain and use energy. They do this by metabolism. Metabolism is a fancy word for the sum of all chemical reactions. And there are two main types of metabolism. Anabolism, which is simple to complex, building up reactions. And catabolism, which is complex to simple, or breaking down chemical reactions. Some living things are heterotrophic, and some are autotrophic. A heterotrophic organism feeds on other things. You are a heterotrophic organism. A plant is an autotrophic organism. It makes its own food. Living things can also reproduce. There are two types of reproduction: asexual, which means it takes one cell or organism to make more, and sexual reproduction, which means it takes two cells or people. The fifth characteristic of living things is that living things respond to their environment through movement, either internal movement or external movement, through irritability, which is the ability to respond to a stimulus. An example of a stimulus could be sight, sound, touch, pressure, chemicals, color, light. And living things are adaptable to their environment. So those are the characteristics of living things. So, each of you think of an example of a living thing and jot it down on your piece of scrap paper. Come up with characteristics of that living thing. Okay, my living thing is going to be my cat, since he keeps walking by me trying to get my attention. And I'm going to list the characteristics of my cat that make it alive. So, for example, my cat, I know, is multicellular. Why else, or how else, do I know that my cat is alive? Go ahead and list 4 or 5 characteristics of your living thing that you pick. Okay, who would like to share their organism and their characteristics? How do you know your organism is alive? Alicia, go ahead.
My animal was a bird, and I said it flies to respond to its environment or stimulus. It is multicellular because it's larger. It lays eggs, which is through sexual reproduction. It grows from a chick to adult, which is from growing and development. And it eats worms or other living things, so is therefore heterotrophic, so it can obtain energy.
Awesome. Great job, Alicia. Who else would like to share with us? Any other volunteers? Remember, participation is part of your grade. Any other volunteers? All right, I will use my cat. My cat, I know, is multicellular. He is moving around, so I know that he is alive. He can adapt to his surroundings. When something is chasing him, he runs away. When my son is playing Nerf darts, he leaves the room. He is adaptable. He does respond to stimulus. If I start petting him, he will lay down at my feet and start purring. And he as well is heterotrophic and eats all of the food that is everywhere. Anyone else want to add to that? No? All right. So now let's talk about taxonomy. Taxonomy is the way that scientists name things and assign them into groups. Very similar to classification, but it's with scientific animals. So why do we have a classification system? Well, a classification system is important for scientists for a couple of reasons. The first is it allows scientists to have one single name for each organism. We in this class are spread across the United States. I'm in the Northeast. There's someone who is, it looks like, in Northern California, or actually Oregon. There are 2 people in Texas and 1 person in New Mexico. We are spread all over. Something that I call a green bean may not be the same thing that you call a green bean. So when we have a scientific name for that plant or animal, it allows us to all know what we are talking about. So it avoids confusion. The taxonomy also allows us to understand how living things are related to each other. So let's take a look at taxonomy. On your screen, there is a picture of an animal. I'm not going to tell you what this animal is, but if you think you know, I would like you to go ahead and type it in the text chat. Now I'm going to give you warning that there are 5 different names for this animal. If you think you know what animal this is, go ahead and put it in the text chat. Okay, a mountain lion. A cougar, a puma. Great job. This is a mountain lion, a cougar, a puma, a Florida panther, or a catamount, depending on where you live. This is a perfect example of why scientists need a scientific name. The scientific name of this the scientific name of this animal is a Felis concolor. So if I said to Robert, he said that they have a lot of them out in Oregon. If I said, hey, Robert, have you seen any pumas lately? He may know what I'm talking about. But if I say, have you seen any catamounts lately? He may look at me and say, I have no idea what you're talking about. But if I were to say to any scientists anywhere, do you have any Felis concolors? If they don't know what animal that is, they can look it up easy enough. And then they would be able to say, yes, we have about 15 in my yard every day. Some examples of other classification systems are in a library, the Dewey Decimal System. In the section of a music store, things may be classified by rock, classic rock, pop, classical. In another science, the periodic table of elements in chemistry. Can anyone think of any other places or types of classification? Anyone else? Any types of classification systems? Alicia, go ahead.
Chapters in a book.
Okay, very good. Chapters in a book. You may have your bookshelf at home sort of classified, either all, like my daughter's bookcase is all chapter books together, all mystery books together, all research books together. People classify things by topic. Some people classify things by size. Taxonomy in biology or in science uses binomial nomenclature. Binomial nomenclature was developed by Carolus Linnaeus. He was a Swedish botanist. There are two parts to a scientific name, a genus and a species. The genus is always capitalized. The scientific name is always either underlined or italicized. And it is in Latin. An example of taxonomy would be for man. The taxonomical name, or the scientific name for man, is Homo sapiens. In English, it translates to wise man. Homo is the genus. Sapiens is the species. Both are underlined, but sapiens is not capitalized. Lots of organisms share a similar genus, but each species has its own species name. So that lends us to the question, what is a species? Well, a species has a couple of characteristics. The first being, it breeds successfully. It can produce its own viable, fertile offspring. It has unique features. These features might be similar to other species, but all of, let's say, the robins all have a red breast. So they are similar to each other, but no other bird has that same red breast. They do have wings like other birds, but they have some sort of unique feature. And they have similar DNA to other species members. So my DNA is not going to match Robert's DNA, but it is similar. Okay, my DNA is not going to match my children's DNA. but it will be similar. There are 7 groups of living things. These groups are called taxon groups. Kingdom, phylum, class, order, family, genus, and species. A good way to remember that, if you have a hard time, is kings play chess on fine green silk. And that's sort of a little play on words to help you remember. Kingdom, phylum, class, order, family, genus, species. The kingdom is the least specific and the largest group, and species is the most specific. A species contains only one kind of organism. Okay, an example of a kingdom is Animalia. A phylum is Chordata. Class is Mammalia. Order is Primates. Family is Hominidae. Genus is Homo. And species is Sapiens. This picture right here is a cladogram. A cladogram helps to classify organisms. It's another way to classify organisms. This is one of the newer classification systems. And the 3-domain system is another newer way to classify organisms. Today, our lab is going to focus on the old way to classify organisms. So what we are going to do now is we are going to take a look at today's activity in our lab book. I need to pause for a second so that I can restart our recording. If I do the whole hour-and-a-half class at the same time, it has a hard time uploading it. So while I am restarting that, I would like you all to open your lab books, turn to page 13, and please go ahead and read the lab introduction. I'm sorry, not page 13, lab number 13. It is called Classification on page 63. Is there somebody who would volunteer to read it out loud since Sarah does not have her lab book yet? Awesome. Thank you, Alicia.